So I'd like to welcome you to tonight's lecture, Pacific White-Sided Dolphins, Our New Neighbors, with Erin Ashe. My name is Jean, and I'm from the SeaDoc Society, and I, along with Joe T. And Laura from Camp Kyla, would like to welcome you to tonight's lecture. This is our 11th season, and the lecture series would not be made possible without our sponsors. So if you see them on the streets or at Island Market in the produce section, please thank them. It's Tom Averna from Deer Harbor Charters, Barbara Bentley and Glenn Prestwich, Barbara Brown, Dean and Audrey Stupke, and West Sound Marina. So please give them a thanks, you guys. Thank you. So here's a really silly question. Joe T and I were talking today, and it's like, how many people do you think in the audience knows how SeaDoc works? How many of you guys know how we work? So we're doing stuff with the marine ecosystem, but do you know how we go about funding science projects? I'm going to tell you. So not only do we do in-house projects, but we do out-of-house. We have a group of science advisories that meet once a year, or depending on how we're doing for funding, to determine what projects they see, because they are from both BC and Washington, state, federal, NGO organizations, and they know from their work what is getting funded and what isn't. So they meet, we pull them together, they get in a room, it's a big kind of like come to Jesus meeting, and who's doing what, who's not doing this, no one is touching this. Most of our projects are funded, the dollar amount is so small that the bigger organizations like NOAA won't look at them, but no one is looking at them, if that makes sense, because they're so small. So then what will happen is we have what's called a RFP, Request for Proposals, and that science advisory will come up with a list of projects. We'll put it out to people, they submit it, their proposal for the projects that we're asking to be, that we're interested in researching. Then we get all those proposals, and they come together again, and they grade those proposals just like a teacher in a classroom. And I remember sitting in on one meeting, and it's like, there's no way we're going to fund this guy. He won't get published because he can't even write a complete sentence in his proposal. And he did a misspelling too. Doesn't he know about spell check? So it's kind of like that kind of thing. So that is how Erin's project came about. No one has been studying. No, she can spell. And no one has been studying the Pacific white-sided dolphins. We don't know, comes to find out that you can ID them just like you do the killer whales. We don't know their movements. We don't know their life histories or their population status. And they're coming more and more. I don't know if any of you guys saw this winter in East Sound, there was some Pacific white-sided dolphins that came through. So it's that kind of work. And our funding comes through private donations like people from you. And what that allows us to do is to be nimble. We aren't a ball and chain to a grant because we have private funding. And then from those findings, what we hope to do is then turn that into policy. So that gets submitted to the legislation in Washington State or up in BC. And also our whole work is to work transboundary. As far as I know, we are the only marine ecosystem organization that is working transboundary. So that's just a little to tell you how we go about doing our funding. So, Erin, I understand she is wicked good at math. And she says not really, that it's more stats, which stats scare the heck out of me. She's a PhD student at University of St. Andrews. And without any, I'm just going to let you tell your story.
Okay.
I'm super excited, so thanks.
Thank you. I wanted to thank Jean very much for having me here this evening. I'm thrilled to be here on Orcas Island. Thank you to SeaDoc for funding this research and for having me here tonight. I've been an admirer of SeaDoc Society for a long time. In fact, I can still remember my first SeaDoc lecture about 10, 11 years ago when I saw Dr. Peter Ross give a talk on the effects of contaminants in killer whales. And I feel in many ways that SeaDoc has been there for me along my career path. So it feels a bit like a homecoming to me coming here tonight. I've spent the last 2 years at the University of St. Andrews, which is in Scotland, at the Sea Mammal Research Unit, doing my PhD. I'm in my final year of about 6 months, so I'm wrapping up. And before I went to Scotland, I was conducting field work in British Columbia on the Pacific white-sided dolphins. My partner Rob Williams and I set up a charity in Canada to support the work that we do there. And I'm delighted to be here to share some of the work and the results that I've found from my PhD. And I mentioned that this is a bit of a homecoming, and that's because one of my earliest, fondest childhood memories is coming to San Juan Island to visit my aunt who lives in Friday Harbor. My mother took us to visit her, and sometime in the wee hours of the morning, I remember being woken up and taken outside being told to listen. And I was very quiet. I was a little young, so it was hard. But I did my best, and I could hear this incredible breathing of killer whales passing by the house. And that memory stayed with me. I was an animal lover practically from birth, and this kind of solidified it. And so I decided very early on, this means I need to be a veterinarian. If I want to work with animals, that's what you have to do. So I went to university to study biology. Around that time, a few things happened. The southern resident killer whale population experienced a pretty dramatic population crash. They crashed by about 20%. And this was what now caused the endangered status, the endangered status of the killer whales. And the other component was that to support my enthusiasm of biology and whales, my mother purchased this book by Alexander Morton called Listening to Whales. So with these two things coming together, I thought, okay, now I know I'll be a wildlife veterinarian, then I can work with killer whales and maybe do something to help out. But then came sort of one of those life-changing moments, and I didn't know it at the time, but looking back, I went to a killer whale conference in Seattle. That's where I met Joe Gatos. I admired Joe, of course, because he was a wildlife veterinarian. He was living my dream, and I went up to him to ask for career advice. And what emerged from my conversation with Joe was really what I wanted to do was be a marine biologist, and that when the time came, if I wanted to investigate diseases or parasites or contaminants in whales, I could partner with veterinarians. And I always credit Joe and think back to that time of setting me on the path where I am now. I also met another influential person about 10 minutes later. I walked across the room and introduced myself to Robin Baird, who is a well-known killer whale biologist who works regularly here in the San Juan Islands. And he hired me my first job as a marine biologist. And Robin then introduced me to my future partner, Rob Williams, who is also a killer whale biologist. So it was this incredible moment that I'm just coming to appreciate now. Rob and I actually both went to work with Robin Baird in North Carolina to study bottlenose dolphins. And when we came back, Rob invited me to come to British Columbia. I don't know if this is showing up very well, but at the very northern tip of Vancouver Island is a little town called Alert Bay on a small island called Cormorant Island, and next to there is Rob built us a small cabin on a little island. So I just couldn't believe that I was going to this incredible place. All of a sudden, the places that I read about in Alexander Morton's book were coming alive. I was in Alert Bay, the home of the killer whale. When you get off the ferry, there's a sign that says that. And it was Mecca. And it's an incredible place because the northern resident killer whales return there each summer to rub their bodies on these smooth pebble beaches. It's a unique behavior, and so unique, in fact, that people came together and decided to protect it, to create a marine protected area. It's the world's first and currently the world's only recognized marine protected area. So Rob picked me up in his boat, and we set out, and we didn't see any killer whales. Instead, we saw this. And from a distance, it looked just like a wall of white water or a river moving through the ocean. And I asked Rob what it was. He said, it's lags. And I had no idea what he was talking about. And I was too embarrassed to ask because we just kind of met, and it was, he's an official marine biologist. And so I just kind of thought, well, it's probably really exciting. It's probably a marine mammal. Let's just kind of hang in there and see what they are. And when I got up close, of course, there were dolphins. And they're called lags because their scientific name is Lagonorhynchus obliquidens. And one of the reasons I was surprised to see them in this location is because it was in inshore waters, much like we are here in the San Juan Islands. But Pacific white-sided dolphins are, you look in any guidebook and it says they're a pelagic species, they're oceanic. They occur in groups of 1,000, sometimes 3,000. They're acrobatic. And what I didn't know at the time was that in the early 1990s, the Pacific white-sided dolphins began to recolonize the inshore waters of British Columbia and the Salish Sea. And recolonize was the key word because archaeological evidence from First Nations middens pointed to dolphin remains that date back thousands and thousands of years. So this wasn't an invasion, this was a coming home. And just to zoom out for a moment, to give you an idea of why they are called pelagic, Pacific white-sided dolphins range from Alaska down to Mexico, across to Japan. So it's true, they are oceanic. And if we look at their distribution and the abundance, you see these colored squares indicate how many dolphins there are in that spot. And surveys from the 1970s and 80s claim that there are 1 million dolphins in the North Pacific. But if you can see the plus and minus 90% there, that's kind of iffy. California did better. Southwest Fisheries Science Center, they are world-class at conducting line transect surveys. They estimate about 60,000 dolphins, plus or minus 50%. And Rob Williams conducted line transect surveys in the inshore waters of British Columbia and estimated about 25,000 Pacific white-sided dolphins, give or take 35%. And Jean was mentioning population status and identifying those species for which we don't have a lot of information to make informed decisions about conservation. And white-sided dolphins fit that description to a T. If you look at the conservation status worldwide for the International Union of Conservation of Nature, it says least concern. The USA, not a strategic stock because there are a lot of dolphins, and Canada has not assessed the conservation of dolphins in about 20 years or so. And sure, taken as a whole, the species looks as though they're doing well. But if we go back to my early days of a budding marine biologist, the killer whales, the resident killer whales here experienced a 20% decline in their population, and they were listed. It wasn't overnight, but it was deemed a critical time to investigate why these whales were declining. And if we look at these numbers, the population would almost have to disappear before we could detect a 20% decline. We wouldn't be able to detect a 20% decline. The dolphins would have to be on the brink of extinction before we would know to act. So we have to come up with other measures of population health. And so we have to start at the beginning. No one has looked at individual biological populations of Pacific white-sided dolphins. And this is a concern because if we remember back to this slide where we're looking at the entire species, that their distribution, they could be divided up into several different individual populations that we know nothing about. And knowing what we know now about killer whale populations, we would never treat them as one species. If you look on the right side of the slide, they all look like killer whales. We all know they are, but they have different eating habits, they have different genetic structure, they have different life history, they have slight differences in even how they look. And before SeaDoc, there really hasn't been any funding or incentive to study Pacific white-sided dolphin structure. So we treat them as just generic dolphins. So going back to my sort of personal story, I was intrigued by the species, but the funding and the incentive wasn't there at the moment. So I went back to San Juan Islands to research killer whales. I was motivated by this idea that the northern resident killer whales had a protected area, and I thought if it can work in British Columbia, it certainly could work here in the San Juan Islands. So I set out to identify feeding hotspots, important feeding areas for southern resident killer whales, so that if the political will is there, we could create a marine protected area that would set aside priority habitat. So we published this paper, and it's available if the time ever comes to create a marine protected area. So soon after I finished this project with the southern resident killer whales, I went back to BC, and I was introduced to my hero, Alexandra Morton. Alexandra, many of you may know her story, but for those who don't, she moved into an area called the Broughton Archipelago about 30 years ago to study killer whales. And during her time there, sort of the late 80s, early 90s, salmon fish farms to grow Atlantic salmon also moved into the area at the same time. And there was some interactions between seals and sea lions coming in and stealing fish. So the farmers responded by putting in sort of alarm devices, acoustic harassment devices, in hopes of deterring seals. And it had the unintended consequence of also scaring away porpoise and resident killer whales and transient killer whales from the area. And the resident killer whales, they removed the acoustic harassment devices, but the resident killer whales have not returned to that area to this day. Along with the fish farms, though, also came dolphins. I don't have a pointer, but so Alex began a photo identification catalog, going out in her boat and taking photographs of dolphins. As the fish farms really established themselves, Alexandra began to notice transfer of disease and parasites to wild salmon, and this captured a lot of her attention and energy, and some of her work with dolphins began to receive less attention. So she knew I was eager to begin research on dolphins, and she gave me her blessing to pick up where she left off with dolphins. And more importantly, she shared her more than 20 years of hard-won data on dolphins in the area. And I say hard-won because this is an extremely remote area. She began this research in the 1990s when we were all still using film cameras. And this required her to take a photograph, go out, photograph all the dolphins, remove the film, put it on a float plane. The float plane would go and deliver a contact sheet. Alex wanted to save money, and because there was no funding for the dolphin research, so she would get a contact sheet back, circle the best dolphin photos, send it back, and then have it delivered, and organize this beautiful catalog. So I'm personally and professionally indebted to her sharing her research with me. So I had everything I needed. I had this amazing data set, and so, Rob and I we had our research permit, we had the boat, we had the dog, we set out. And we saw all sorts of things. We saw grizzly bears, killer whales, of course, stellar sea lions. It's an incredible place to work. There's a harbor porpoise. Last December, just a few months ago, we saw a gray whale. There are fin whales, humpback whales. You get the idea. But driving around in a boat looking for dolphins, taking pretty pictures isn't science. So I had to develop some research questions. And I came up with an acronym for my study and outlined some objectives. To estimate the abundance of Pacific white-sided dolphins, estimate a survival rate using both Alexander's data and data that now is up to 7 years for me, and use this information to assess population health and status. Also to explore population structure using their acoustic repertoire, and I will touch on that a bit later as well, and identify factors, what's influencing dolphin survival, what do we need to be concerned about? Bycatch and fisheries, killer whale predation. But it was all centered around photo identification. And many of you know, with living among southern resident killer whales, that each whale is individually recognizable. They have natural markings that tell you who they are, and this happens to be L78. This kind of research was pioneered by a researcher called Mike Bigg in the 1970s, and it was in response to the live capture fishery that was going on. At the time, we thought there were killer whales everywhere, or thousands of them, so people would apply for permits to capture them and put them in aquariums. And the Department of Fisheries and Oceans in Canada became concerned and asked Mike Bigg to go out and count whales, essentially. A lot of this early work was compiled in an International Whaling Commission special issue, edited by my now PhD supervisor, and it has amazing classic papers by Mike Bigg, John Ford, Ken Balcom. The important thing here is that Phil Hammond pioneered the statistical methods used to analyze cetacean photo ID data. So once you have a photograph, how do you turn that into actual data that you can use to find out how well the population is doing? But Phil Hammond works at the University of St. Andrews. So I traveled from home of the killer whale to the home of golf, which seems a bit odd to people when I first tell them. But the University of St. Andrews, the Sea Mammal Research Unit, has an extraordinary partnership and collegial relationship with some world-class statisticians, and that's the sort of training that I needed to study Pacific white-sided dolphins in this context. And that's because you can't count every dolphin. Unlike killer whales, not every dolphin is well marked enough to follow it through time. And there are many, many more of them. So the likelihood that you'll go out and capture every single fin is low. So that's where the scary math enters the equation. And some of this capture-recapture statistics or mark-recapture statistics were really initiated by Pierre-Simon Laplace in France to figure out how many people there were in France in 1802. And the basic idea is you have the natural markings and you have a record of individuals that you follow through time. And from that sample, you can estimate these important population parameters like survival or abundance. So here's an example of a classic photo ID photograph of a Pacific white-sided dolphin. You can see, I think, that he has some nicks on the trailing edge of his fin, and it also has some tooth marks from other dolphins, and that's quite common to see these scratch marks. But really we rely on these nicks because the scratches can change over time. So you need to pick a really reliable marking. and these seem to do quite well. So I'll walk you through a quick photo ID day. So let's say you go out on the water, and the first day you capture these 5 dolphins and 10 others. The next day you go out, you capture another 5 dolphins plus 20 others. I'm saying capture, I mean on film, not literally capturing them. And you find, oh, these 3 match up. So I think this is the only equation. This is a very simple equation that you can use to estimate abundance, and you by taking the number of dolphins from the first encounter times the number of dolphins from the second encounter, divide it by the number you saw, the number of matched dolphins from both encounters, and it gets more complex from there. But here are some preliminary estimates from Alexandra's catalog. And you can see there are about 1,500 dolphins that used the Broughton Archipelago region in 2002. It looks as though the population is just going crazy and increasing. And in fact, it's about 10%. But looking at that number, it looks biologically impossible. And it might be. It might be that dolphins from offshore or from other areas are moving into this area. And that's one of the things I'm teasing apart now. You can see that calves, another interesting aspect of this is that baby dolphins weren't seen in the area until 1995. So that also lends itself to the idea that dolphins are moving into this area. And there may also be some population growth too. So that's, I'm trying to find out how much each part is contributing to this growth. So just a quick look at the babies that are coming into the area. Again, they were not observed in this area until 1995, but now we see them regularly. They're every summer, and they're in some of the same places. So it's an interesting phenomenon to watch. And we suspect that some of the dolphins are moving into this area, and it's probably because of food. Their diet consists mostly of herring. Kathy Heisey did an incredible study in the late 90s. Alexandra Morton has done amazing studies as well, looking at diet, and found a seasonal pulse. So herring, then they shift to capelin. Ooligan as well is an interesting one that we're looking into. And we've done opportunistic studies, and we hope to do more intensive diet research. And I have a little video here just to get a sense of how they forage for herring. And I've slowed it down so you can get a sense of their movement. So it's really interesting to think of the level of coordination and mind power that must go into corralling fish like this. So there's a lot left to learn about that particular foraging strategy. And when the dolphins are engaged in that feeding behavior, it is lightning fast, and it's not conducive to photographs. So I usually sit back and videotape it if I can, or collect prey samples, ideally. And back to the photo ID aspect of the study. Important part of photo identification, apart from the fact that it's non-invasive, it can tell you about population trajectories, can also tell us important things about movement. So are the dolphins using the Broughton Archipelago and the Salish Sea equally, or Georgia Strait more specifically? So it's both, tells you something about how the dolphins are moving both in time and space. This summer, for example, we had an 11-year recite of D986, and it was photographed in the exact same spot that Alexander photographed him 11 years previous. So that tells us something, that dolphins are using this habitat on a regular basis. They have some sort of residency, or at least some sort of site fidelity. We also have matches between dolphins seen further south, here in the Salmon Islands, and the Broughton Archipelago. So and these are the first bits of information like this that we have for these dolphins, and with respect to movement patterns. Going back to population structure, early results from the photo identification tell us that dolphins aren't doing as well as we expected when you compare to other similar dolphin species. So that was a concern. But we just have to find out, does this apply to all of the dolphins across the Pacific, or here just in British Columbia and the Salish Sea? We have genetic evidence to tell us that they do divide themselves into small populations. In Mexico, for example, they have different skull shapes. You can't see it from on casual observation, but it is there. And again, we have historic matches between Washington and BC. So we're hoping the acoustics can tell us something. And I was hoping to play this sound, but obviously I'm having to shout, and I don't think my computer can do much better. But this is an example of a dolphin spectrogram, and they're very chatty. And this can tell us something about who they are and which population they belong to. As I mentioned, survivorship is lower than we thought. And if that's true, we're not, I'm still unraveling that piece a little bit because I was surprised by the result. But if that's true, one of the first things I will look at is fisheries bycatch, because we know dolphins in the Pacific white-sided dolphins specifically were vulnerable to bycatch in the 70s and 80s. Hundreds of thousands were killed in the high seas drift net fishery, which is now fortunately banned, but some drift nets are still in use illegally. Pacific white-sided dolphins are hunted. There's a quota, an annual quota in Japan to drive them into the bays and collect them. The survivors are often sold to aquariums, and the others are often sold for meat. A colleague of ours, Scott Baker, who works at Oregon State University, has developed this incredible tool. It's a portable DNA lab. So Scott will go to fish markets in Japan and Korea and test the meat that's labeled minke whale meat. And often he's found out that it's actually Pacific white-sided dolphins. Here in Pacific Northwest, Rob Williams and colleagues conducted a SeaDoc-funded study to investigate bycatch, and found that dolphin bycatch does not appear to be a problem here. For porpoise, it is, but the observer coverage in dolphin habitat was a bit scarce, so we're still kind of leaving that open for investigation. And here's just, bycatch does not appear to be a problem, fortunately, in California, and it's unknown currently in Mexico. This dolphin has lost its dorsal fin, and we see evidence of interaction with some kind of fishing on the dolphin dorsal fins, but we're not sure exactly what it could be from. We do have local reports from speaking with fishermen that they've had dolphins entangled in their gill nets. And there have been some rare instances of dolphins caught in seine nets. And we've had these reports of entanglement in fish farm anti-predator nets. So when the acoustic harassment devices had to be removed, fish farmers had to do something else to keep seals and sea lions out, and they've put up sort of a net that creates a wall and barrier between the seals and the fish. And sometimes a farm is abandoned and it's left loose, or human error, a net is left loose and there are casualties, often sea lions. I hope to play a video, but I don't know that the sound will work. This is a sea lion caught, obviously, in an anti-predator net. And at the same time, there were reports of dolphins being caught in this net that had been left, that was abandoned. And there's no law against that currently. And we don't really know to what extent this is happening and what impact it could be having on dolphin or sea lion populations, for that matter. But it may not all be due to human interactions. We see killer whale tooth rake marks on dorsal fins, on the flanks of dolphins. So these are obviously the survivors. And I have another video here of white-setted dolphins. This is a typical day out on the water. It's actually a really nice calm day. This is in Night Inlet, and I did have audio again, so there are dolphin vocalizations going on here. We were with a group of about 300 or so. And then we started seeing this behavior of a dolphin leaping onto its side repeatedly, like 30 times maybe. And we thought something was up. And then we saw the dolphins collect and swim to shore as quickly as possible. And we thought, we've done something wrong, because they're freaking out. And then we found out why. There was a pod of killer whales, and we were duped. We've studied killer whales for 10 years, and we had no idea they were there. The killer whales slowly herded all of the dolphins into a shallow, enclosed bay. And this one sadly didn't make it. This was my first day in the field as an official PhD student, so I was kind of blown away by that. But we managed to collect the heart and lungs of that dolphin, so we'll have some genetic information about it. So we know that this kind of predation is going on, and I'd like to find out more about how often it happens and what it's like living with that predation risk. Because we know even living with the risk of predation can cause stress, and that has an impact on survival. And we know that dolphins are living with the risk of killer whale predation about 20%, at least 20% of the time from our early analysis. So the next steps for me are to complete my photo ID analysis and unravel some of these remaining questions, to investigate further the killer whale predation aspect, and complete the acoustic analysis, and to continue this dolphin project, because long-term data sets are extremely valuable, irreplaceable, some may say, and we have a lot to learn. So there are incredible future opportunities to look at stress hormones from dolphins, and to really address the anti-predator nets. I showed you the video of the sea lion and the possibility that dolphins are also getting caught in those nets. Fish farms bring in a lot of disease, and we're curious as to whether any of that transfers to dolphins, and combine some of the genetic evidence that we have with the acoustics, to learn more about how the dolphins are divided into different populations, and to collect more photo ID photos here in Washington and more recordings. So with that, I'd like to thank all of you for your time and attention, and to SeaDoc and all of his supporters. Thank you very much. Sure. I think we have a few minutes for questions if anyone has. Yes.
Do you know if killer whales have always gone after the white-throated dolphin, or is this a new behavior because of other food that's less accessible for them?
There are reports from this area in particular dating back to the early 1990s, and it's likely that that sort of predation has gone on for much longer previous to that time. But it's whether it's reporting or a trend, it does seem to come in pulses where you'll see there will be reports quite close together. You'll get 3 or 4 reports in a month of killer whales coming in and attacking dolphins. So I'm not sure if that's a real pattern or it's just due to having lots of people out on the water getting really interested and attuned to it. Yeah. Yes.
I was curious, Erin, do the Pacific white-sided dolphins hunt, do they do the blow bubble nets the way the bottlenose do?
We don't know. That's something that would be fascinating to study. The video that I showed you of the dolphins corralling the herring is an extraordinary behavior, and I can't wait to learn more about it. I think we may have an opportunity in the fall to have some support with an underwater camera. So I'm really looking forward to that. Yes. Oh.
How long have you been doing this research? And you said you were up in the Lord Gray area at Johnston Strait, and then obviously you've done some work down here.
Yes.
So have you done the whole inside passage area, or what?
For the dolphin research, it's been mostly in the Broughton Archipelago area, Johnstone Strait area. I've done killer whale work down here, and then with Rob, I've done some work in the inside passage, both looking at estimating abundance of marine mammals in BC, and more recently we've been collecting information on ocean noise. We've deployed large autonomous hydrophones, 12 of them all up and down the inside passage to collect information about ocean noise.
How long period of time?
In my lifetime? Or
How long have you been doing the research on the dolphins?
on the dolphins. I began my research on dolphins in 2006, something like that. Yeah. Yes.
In this area, how many dolphins are seen in a group?
Last time there were about 300, is from what I hear and what the photographs I saw. That looks like a good estimate. I received some photographs from Robin Baird's research group. They took some ID photos for me, and that was quite a small group, just I think about 5 dolphins. So it's quite variable. Yeah. Yes.
Yeah, the distribution pattern you had early on, it just shows them that they're in the North Pacific,
Yes.
and why is that just because of their preferred food, or?
That is how, yes, that is how they evolved over time. They have a cousin in the southern hemisphere called the dusky dolphin, and they look almost identical. And
Like this latitude, south and north?
I guess, yeah, they're in New Zealand and Australia and yeah, well, yeah, but they're kind of more distantly related.
But they're also temperate.
Yeah, that's true. And yes, sorry. The genetics, the taxonomic story is evolving quickly with white-sided dolphins, so they may be assigned to a new genus and species. So I'm, yeah.
The confusion could be that the blue is the range and
Oh.
the colors are places where we have data.
Oh, that could be.
Was that clear? That the blue is the range and the colored dots are the places where we have data where people have ever surveyed, and the shade of the color is the density. So So you can see there are huge chunks that have never been surveyed.
So we expect them to be in those areas, but we've never looked.
We need a bigger boat than our 18-foot boat.
Yes. Yes.
Erin, why St. Andrews? Are there cetaceans up there?
There are. People may be surprised to find there's actually a great population of bottlenose dolphins there that are quite well known, much like our southern resident killer whales, and a lot of great work has come out of there. But I think for me, both the supervisor in particular was well suited to what I wanted to accomplish. And the interaction between the statistics department and the biology department is like something I've never experienced. It's incredibly collegial, and it really leads people to the cutting edge of science. So it was a good choice for me for this project. Yeah. Yes.
So they're much more common up in Canadian waters than they are down here. Like that group of 300 is sort of unusual to see.
Yes,
in
but
Canada down here.
yes.
And you mentioned that it's a reintroduction there actually because they were found in the midden sites. Do you know about the midden sites down here?
I don't. I've done some looking in. I've looked into that a bit, but I'd like to look into that more because I'm trying to remember if there may have been evidence in one of the midden sites down here, but that is something I'd like to investigate further because that would be really interesting. Yeah.
I'm just wondering how they get those nicks in their fins that you identify them by.
A variety of things. It can be injury or the way the fin grows or interactions with one another, life.
Those would change if they acquired them in such a long succession.
Yes, that is true, and that's something you definitely have to watch out for. That's why it's very important to have high quality photos and be very strict. But it's somewhat like if you take a photograph of someone over years and years, maybe they change their hair or they gain weight or something. You can still identify that is that particular individual. But that is something to definitely be careful of, that you're not assigning an incorrect identity.
It must be really hard to be that familiar with these critters and then suddenly become lunch.
Yeah. Yes.
Do they have acoustic signatures?
That's something I'm finding out at the moment. I'm in the middle of processing the acoustic data right now. Yeah. But they don't have whistles like bottlenose dolphins. They are more broadband in their vocalizations. So I'll do this and then Erin. Yes.
What's the lifespan of the dolphins around this area, SAHC? What's the mortality rate?
The mortality rate that I've estimated is about 12%, which we think may be a little high, and so I'm investigating that further. And they live to be about 30, 40 years. Yeah. Oh, Erin.
Do you have any underwater footage, kind of like the Orca lab? Or is there still camera just ready to capture imagery?
No, but they do swim past Orca Lab. Yeah, so if they could just stay still. They're yeah, they're kind of like killer whales on just fast forward all the time, and they change their behaviors quickly. They'll be resting for 30 seconds, and then they'll forage, and then sometimes you do get long bouts of a particular behavior, but it's certain the cameras are available for them to swim past if they would do it, and they are commonly spotted around Orca Lab. Yeah, and they're great at tipping me off when dolphins are in the area and recording their acoustics for me. Yeah. Yeah. Yes.
So around here, we usually have seen water porpoise over the years, and they always have small groups, like three animals, maybe as many as 11, and that's kind of it.
Mm-hmm.
What would be the difference in their, the prolificness of one population having 3,000 or 300, while another one stays in like these little family groups of 11 or 7 animals? I realize you probably don't know because you haven't studied that long, but any ideas why like a
Yeah, it's interesting. In the Broughton, sometimes we do see groups of 11 or 25, and that's kind of around mating season, and then they do tend to be off on their own. And it's likely related to their social structure and the food that they eat and their strategies for making a living. The harbor porpoise are very high frequency callers, I guess you could say, and they have different foraging specializations than white-sided dolphins do. So that could be playing part of it. Yeah.
Okay. All right. Thanks.
Thank you
Erin,
very
so
much. Thank you.
So, our next lecture, our last lecture for the season is March 12th. Dr. Milton Love will be here. He's very entertaining. And also for housekeeping, don't worry about picking up the chairs. We got you covered.
Wow.
All right? Thanks.
Thank you.